You're welcome to Excellent Simplified Tutorials.
Alright, so we are going to take some questions on nucleic acid. So the first question here says, with the aid of diagram only, differentiate between the purine and pyrimidine ring structure. I take that again. With the aid of diagram only, differentiate between the purine and pyrimidine ring structure. Now, that question emphasizes on purine and pyrimidine with the aid of diagram only. One thing we should note is that the purine is a fused ring, two rings combined, while the pyrimidine is a single ring. So the simplest way to draw a purine ring is carbon, 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 nitrogen, carbon, nitrogen, carbon. This is the first ring. The next ring, nitrogen, carbon, nitrogen, carbon. This is your purine ring. As you can see, it's actually a fused ring, a 6-membered ring and a 5-membered ring. Now for our pyrimidine, pyrimidine is a single ring, a 6-membered ring. And to draw that, carbon, 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 nitrogen, carbon, nitrogen, carbon. This is your pyrimidine ring. So they said using diagram only, differentiate between purine and pyrimidine. This is what you're expected to do. This is the structure of purine and this is the structure of pyrimidine. Now, if that question have asked to differentiate between purines and pyrimidine using specific examples, the examples of purine are adenine and guanine. These are the two basic examples. Then the examples of pyrimidine are cytosine, uracil, and thymine. Cytosine, uracil, and thymine. So adenine and guanine are two examples of purine. If you want to remember that, you can use pure as gold. Pure as gold. Pure for purine, as for adenine, gold for guanine. Then for the pyrimidine, if you want to remember the basic examples, you can use CUT, C-U-T. C-U-T. Where C is for cytosine, U is for uracil, and T is for thymine. If you want to draw the structure of adenine, now this is the basic purine ring. But if you want to draw the structure of adenine, the only thing you do here is add amino group here. This is your adenine. If you want to draw your guanine, what you do is the amino group comes here and you have a carbonate carbon here. But this is not required in the question. I'm showing this in case of another question. But basically that question, you are required to draw just this basic purine ring and the pyrimidine ring. That's that. Now the next question there says, why is the biosynthesis of purine and pyrimidine critical for all cells? Why is the biosynthesis of purine and pyrimidine critical for all cells? Now this is our purine and this is our pyrimidine. The question is asking, why is it critical? Why is it important for all cells? Now let's look at nucleic acid. I'll use that to explain this. Nucleic acid is divided into two. We have two types of nucleic acid. We have the DNA and the RNA, right? Now the building block of this nucleic acid is called nucleotide. Nucleotide is the building block of nucleic acid. Nucleotide. And the nucleotide is composed of three things. If those three things are not there, you don't call it nucleotides. So the three things found in the nucleotide are: number one, nitrogenous base. Nitrogenous base. Number two, pentose sugar. Pentose. And number three, phosphate group. Phosphate group. Now these are the three things that makes up our nucleotide: nitrogenous base, pentose, and phosphate group. Now if you keep this phosphate group aside, that is, if you remove this phosphate group, what you are now having is no longer a nucleotide but a nucleoside, because you've kept the phosphate group aside. So if you keep the phosphate group aside, what you are having is now a nucleoside. So nucleoside contains only nitrogenous base and pentose sugar. Now the nitrogenous base here can either be a purine or a pyrimidine. A purine or a pyrimidine. We are coming back to this now. So you can see that we said that nucleic acid, the building block of nucleic acid is our nucleotide. Nucleotide is the building block of our nucleic acid. And now we are seeing purine and pyrimidine as part of the building block, as part of the nucleotide. That means without purine or pyrimidine, there will be no building block for nucleic acid. And as human beings or animals, we are multicellular. We have cells all over, and nucleic acids are found in these cells, in the nucleus of these cells, which helps in the transmission of genetic information from generation to generation. Now, if there is no purine or pyrimidine, nucleic acid cannot be synthesized, and so genetic information cannot be passed from generation to generation. And therefore, purines and pyrimidines are essential, are critical for all cells. So that's simply the answer to that question. Now, if you are to go on, the pentose here can either be ribose or deoxyribose. Ribose or deoxyribose. And just like we said earlier, that the two types of nucleic acid we have are DNA and RNA. DNA and RNA. DNA is deoxyribonucleic acid, while RNA is ribonucleic acid. Now, ribose is only found in RNA. That is why it's called ribonucleic acid. Nucleic acid containing ribose sugar. Nucleic acid containing ribose sugar. So ribose is found in RNA, while deoxyribose is found in DNA. That is why it's called deoxyribonucleic acid. Nucleic acid containing deoxyribose sugar. Good. Now, we've seen that purines, the examples of purines are adenine and guanine. Adenine can be found in DNA, can be found in RNA also. Guanine can be found in both RNA and DNA. But the problem comes here in uracil and thymine. Uracil can only be found in RNA, while thymine can only be found in DNA. So that's the basic difference. In case you are asked for the difference between RNA and DNA, the sugar found in RNA is ribose, the sugar found in DNA is deoxyribose. Another difference, the base that is peculiar to only RNA is uracil, while the one peculiar to only DNA is thymine. If you have any question, please drop it in the comment section. I will respond as soon as I can.